Hello, my name is Alecia and this summer I participated in research at the Children's Hospital of Philadelphia through the assistance of PURM funding. I had the opportunity to work in Dr. Johnson Gupta's lab, focusing on the effects of circadian regulation on lung injury and repair. In this lab, I worked closely with Dr. Andrea Paul, researching the effect of environmental light disruption on circadian-gated lung injury. The human body's circadian clock plays a critical role in regulating our immune response, particularly during infections like influenza. Our research delves into how disruptions to this internal clock, specifically through environmental light changes, can exacerbate lung injury and inflammation during influenza infection. Studies in mouse models show that the timing of an infection can significantly impact the severity. For instance, infections occurring during the body's active phase result in a higher viral burden and more severe lung inflammation. Prior research conducted in this lab focused on the core circadian gene BML1, which, when disrupted, makes the body more vulnerable to environmental disturbances and worsening influenza outcomes. However, it has been found that introducing food cycling as an external cue can mitigate these adverse effects. A real-world parallel to this disruption is the environment in intensive care units (ICUs). However, despite their role in patient recovery, ICUs often expose patients to constant artificial light, disrupting circadian rhythms and potentially worsening outcomes during infections. Our research aims to model this disruption and study its impact on lung health during influenza infection. To do this, we conducted a series of experiments to test our hypothesis that environmental light disruption exacerbates lung injury during influenza infection. We analyzed bronchoalveolar lavage, BAL, fluid to quantify immune cells like macrophages, neutrophils, and lymphocytes, revealing a marked increase in immune response and viral load under disrupted light conditions. RNA sequencing and pathway analysis further identified differentially expressed genes and pathways linked to inflammation and injury, with the most severe effects observed in the constant light group. Histological analysis of lung tissue also confirmed these findings, showing that mice exposed to constant light post-infection experienced significantly worse lung injury than those in controlled light conditions. Additionally, high levels of CD3 markers indicated an increased presence of inflammatory lymphocytes, further driving lung damage. In conclusion, our research highlights the significant impact of circadian rhythm disruption, specifically through environmental light, on lung inflammation and injury during influenza infection. These findings underscore the importance of maintaining circadian stability, especially in environments like the ICU, where constant light exposure could worsen patient outcomes during infections. Our next steps involve further exploration of the critical pathways and critical genes responsible for these effects, with the potential to inform new therapeutic strategies to mitigate the impact of circadian disruption on health.